you do get it back, so far we're batting almost 100%. I'm getting them back. Not quite 100%. But you get data like this. This is the growth rate of the alkanone producing haptophytes based on that rate of labeling. Okay, so as well we're showing depth. So this is light. Guess what? If you go deeper in the ocean, there's not as much light. So it gets darker. Growth rate slows down, as you would anticipate. Less light, there's less rate of growth. There's a light level for virtually all plants below which light is limiting. And for the alkanone, well, at least for Huxleyi, this thing called saturating irradiance, people argue about it. The range is something like 72 to 1,000 micro-Einstein. Here we got 400 at about, what? Pretty close to 5 meters. So if the saturating irradiance was 1,000, all of these would be light limited. But, you know, here's some at 100, 72 to 114, you know, 120 to 300, some around 80. Well, when we have data like this, we have light and you have growth rate, you can cross-plot the two. And look at that. You increase light, you increase growth rate. This is evidence that the rate at which those cells are grown are light limited. Okay, so that's one implication. Well, one implication of that is that, you know, all those experiments that we would like to test with carbon isotope fractionation data from these compounds, they're all grown at saturating irradiance. You know, you're in the laboratory, you want these things to grow, you give them all the light they want. But in the ocean, that's not true. So we go back, you know, that data point is here. So by about 30 meters, all these depths cells are light limited. Okay, so the other implication is these are haptophytes, so there may be a mechanism by which they transport CO2 into the cell that's dependent upon light. Okay, so, you know, you do those same experiments in the North Pacific and Bering Sea, Gulf of California, subtropical North Pacific, Station Aloha, you get a relationship where the inflection point is around here. It's around 100 micro-Einstein per meter squared per second. So that's the saturating irradiance. So the point is that we have some data points where we can measure, we have growth rate, we can measure the carbon isotopic composition of alkanones at depth. We have the growth rate, we can measure the CO2 concentration in the water, and we can look at carbon isotope fractionation versus growth rate over CO2 under conditions where presumably the cells are nutrient limited. So here they're light limited. Here they're limited by some nutrient, probably nitrate. Okay, well, this was a scare for us. In the Guaymas Basin, there are both Emiliani Huxleyi and Gephyrochapsa oceanica that coexist. And their concentrations vary with depth. So here, in the surface waters, Gephyrochapsa oceanica dominates. And below about 10, 15 meters, Emiliani Huxleyi dominates. Well, the scare was, look at these data. There seems to be a step change where the abundance of these algae change. And the difference is on the order of about 3.5 per mil. So that's 3.5 per mil. That's quite a bit. Here's a scanning electron micrograph of Gephyrochapsa and Emiliani Huxleyi. And so, you know, you have to get rid of the water so they're sort of dehydrated. But, you know, they're about the same size and shape. So it's unlikely that this difference would be explained simply by surface area to volume. There might be a little bit of difference, but you know, they're spherical. You have to change a spherical cell a lot to change its surface area to volume ratio. Okay, so what else could that be? It could be this weird vital effect, which is that explanation given by geologists when they don't understand the biology. Or it could be light. You know, as we get deeper in the ocean here, light goes away, gets it's less light. Well, we can test that. There is an effect of carbon isotope fractionation as a function of light that's documented in the literature. And here's fractionation versus light. Photon flux density, micromole per meter squared per second, which is the same as micro Einstein per meter squared per second, so around 20 to 160. And here we see that fractionation decreases with lower light. Now, one explanation is that as you diminish light, you change that ability of the cell to actively transport inorganic carbon into the cell. So you're changing the flux in, flux out. There's less flux in, there's lower fractionation. Okay, because you're changing the CO2 concentration in the cell. Okay, so at all of these sites, there's variation in the abundance of these two forms of alkanone producing, these two genera that produce alkanones. And so we would expect, if there is a systematic relationship with the dominant species, we could pick it up by comparing these two, these regions. And if it was some vital effect that we don't understand, we would not see consistent relationships between fractionation and light. Turns out we do. We see a consistent relationship. So the difference we saw here is probably due to a diminishment of light and diminishment of this CO2 transport. So what we see is about 3.5 per mil variation from about 20 to 160 micro Einstein, 20 to 160 micromole, same micro Einstein per meter squared per second. So the effect of light on growth, we know that you diminish light, you diminish the growth rate. So in essence, what we're looking at is depth here. So remember the culture results when we plotted epsilon p versus growth over CO2, we saw a slope that was negative. Well, you know, if we go deeper in the ocean, we diminish light, we really are decreasing growth over CO2. We know growth rate decreases with depth, and because of respiration, CO2 increases as a function of depth. So some variation in the isotopic composition of these alkanones is due to an effect of light limitation, and it's probably due to how light affects the transport of CO2 into the cell, that CO2 flux in and flux out. So one of the nice things about these experiments and measuring at different depths is that with this in situ 13C labeling, it yields a growth rate, but it also allows us to identify where light-limited growth occurs. So, you know, we can measure the delta 13C value of the alkanones. We can measure the delta 13C of total dissolved inorganic carbon. We can measure things like alkalinity and DIC concentration, so we can describe the whole carbonate inorganic carbon system and calculate the isotopic composition of aqueous CO2 and determine its concentration. So we have all the information we need to plot epsilon p versus growth over CO2 and compare the field results with laboratory results. And this is what we get here. So, you know, it's somewhat anticlimactic. You know, if you sort of draw a line through your mind here, a lot of the data clusters along this linear trend. One thing it tells us is that really these points out here, based on the experimental results, are irrelevant for nature. We just never find CO2 concentrations that low and growth rates that high for the alkanone-producing haptophytes. And while they sort of cluster along a trend that falls between these experimental data and these experimental data, there's some that fall below. These are from that cruise from hell, that high nutrient, low chlorophyll region. We don't know, but we suspect that potentially the CO2 transport mechanism may also have a requirement for some metal, and that's affecting the ability of the alkanone-producing haptophyte in these environments from fractionating like these do, so that the effect of CO2 transport on that flux in, flux out has some effect on carbon isotope fractionation. What we can conclude from this is some of the major factors that control fractionation. Just like terrestrial plants, it's flux in, flux out, but the details are different. Certainly the concentration of CO2 and growth rate, but also cell geometry affects it. By studying the isotopic composition of alkanones, we can limit cell geometry. We can probably limit isotope variations in all carbon-fixing enzymes. We might be able to limit the amount of bicarbonate and CO2 assimilation, but not all of it. And certainly this 13C labeling, which gives us growth rate, can identify where light limitation of growth is a major control on not only the growth of these algae, but also carbon isotope fractionation. But there are other factors that we still don't understand on what controls growth rate, what controls CO2, and its effect on carbon isotope fractionation. So while long-term variations in carbon isotope fractionation through geologic time will give us a rough idea of what CO2 variations are like, they're unlikely to give us accurate CO2 concentrations in the past. At least right now, until we begin to understand some of these other factors that can affect the isotopic composition of marine microalgae. And you know, this record can go back only as long as alkanones have been produced within the geologic record, which is somewhere around the Cretaceous. I think there are some coccoliths found in Jurassic age rocks, but I don't think there are any reports of alkanones. But they are certainly present in the Cenozoic, so we can produce a carbon isotope record, and it has been. I think Kate will talk about this in her lecture on Friday. We can produce a carbon isotope record of alkanones over the last roughly 50 million years, and it can be related to gross changes in CO2 concentration through time, but getting absolute values of CO2 concentration from these is probably inaccurate. It probably can't be done. Okay, so yeah. Questions about that? That's a lot to digest. I think it illustrates how we can go about in the field making these measurements and constraining some of these factors. So we can make logical interpretations of alkanone variation in the past. Okay, let's take a break there. Go ahead. Well, you know, that's an interesting thing. Another interesting thing about these alkanones, that remember they produce these variable numbers of double bonds. It turns out that the ratio of unsaturation is sensitive to temperature. So yes, you can determine temperature from alkanones. It's the alkanone unsaturation index. So another advantage of using these is simply to use the ratio of the concentration of the di and tri-unsaturated ketones as a temperature indicator.
So
yeah.
When you're looking in the geological record, you look for those two species?
Well, the compounds produced from those two species, okay. Correct. Yeah, or similar species. Actually, these species have only been around for the last, I think, 150,000 years, but there have been alkanone producers that have existed back 40 million years ago.
Oh, so before we even know what produced those alkanones?
Yeah. But at least we're seeing consistent data from the two that produce it today, Gephyrochapsa and Emiliani Huxleyi in the open ocean. But yeah, we don't know what species produce these alkanones in sediments older than 150,000 years. But records for isotopic variation in alkanones exist at least from about 40 million years to present. Kate will talk about some of that. Friday. Yeah?
So
So,
Growth rate
yeah,
is different when you're light limited versus light unlimited.
yeah.
And therefore the fractionation is different.
Well,
Is it
it's the control on growth rate. You know, a number of things can control growth rate.
but
It's,
the growth rates are different in the light limiting zone versus the
yeah,
light unlimited
yeah.
zone. So therefore the C13 would be different. But there's not zero growth in that light limited zone, correct?
It gets, well, yeah, below a certain light level. No, they're not going to grow anymore.
Yeah, well, I was just wondering if that zone that's not optimal but might still be producing some signal, if that there's mixing between those, if that can be.
Yeah, you know, I sort of struggle with that because if you have a concentration and you have a rate of labeling, you've got a production rate of that compound. And normally, production rate kind of follows that growth rate curve, so most alkanones are produced in the surface, well-lit surface oceans. So you would expect where there's greatest production, there's greatest export. And that's what's measured in sediments. So there's diminishing amount of production with depth. So serendipitously, light limitation might not make any difference in the geologic record. But we also find some regions where that production rate is low in the surface, it kind of goes up and there's a subsurface maximum and then it drops off again. And so, I don't know. I don't know what the mix is in terms of what actually falls out of the photic zone or what falls out of the surface ocean and gets recorded in sediments is from alkanone production under light limited conditions. And if it is light limited, we have to begin to worry about how light affects fractionation to interpret that record. It's another complicating factor. But yeah, good question.